Time you going to start? Vincent van Boon, Napoleon Bonaparte. Oh, I like those Napoleon Bonaparte. That's a good one. I need a little one of those like tri-corner hats. If you can find that, that'd be great. Someone had a fracture in their skull, and doctors put the fracture piece in the abdomen to heal. The only reason I can think that might work is because it's easy to access and you can get blood flow to it, but that's very interesting. I'm not entirely sure, actually. Okay, we're gonna get started. Welcome back. Excuse me. Okay, so we're gonna get to this guy in a second. This is a actual skull that's been cut and saturated. Let's first go to those last few slides. Cerebellum. Hey, so I think the last time we left off on what the cerebellum does. It's important for coordination. It's important for balance. Those really highly branched parts that we see here in the cerebellum, those are known as arbor vitae. They're formed because there are lots of dendrites to receive sensory input. The sensory input that we're getting includes vision, somatosensation, touch, balance is vestibular sense, hearing. You can use all these different things for maintaining balance. So we're getting all that input to help us balance. Our cerebellar cortex, the outer portion of the cerebellum. So this outer gray matter, that's cerebellar cortex. That's subconsciously coordinating movement, sending output through the pons to other places, like your pons and cerebrum. One identifiable feature, this isn't the best model for it. One identifiable feature, this is the posterior view, so this is the superior view, dorsal. One identifiable feature is this midline lobe. That's called the vermis. The vermis is a worm-like looking thing on the dorsal side. Vermis. Vermis means worm. Yeah, you can see the vermis right here. There are different lobes. There are different names for connections between the cerebellum to the rest of the brain. I'm not going to test you on those. They're called peduncles. Peduncles are the connections between the cerebellum and the rest of the brain. But you can see how close it is to the other parts. Cerebellum's here. Here's the midbrain with those 4 bumps. Here's the pons. Here's the medulla. Cerebellum is important for coordination. On a gross anatomy level, you can see how branched those things are in the cerebellum, the arbor vitae. And also at a molecular cellular level, we can see the branching. Remember the Purkinje cells? This is if we see a Purkinje cell with really good staining. You can see here's the cell body, and all of this, these are all dendrites. What we saw with our light microscopes, these are Purkinje cells. Purkinje cells with the branching dendrites. Purkinje cell with branching dendrites. It's colored purple here. On ours, it was colored like brownish. So again, don't go by color, go by pattern. Have a cell body in a line, cell bodies in a line, and they branch out in one direction. It's the Purkinje cells that are sending, they're receiving the sensory information, and then they send output on what your body should do to maintain balance, to maintain coordination. Purkinje cells are the cells that you find in the cerebellum, the highly branched cells that we find in the cerebellum. Pyramidal cells you find in the cerebral cortex. Purkinje cells you find in the cerebellar cortex. So pyramidal cells, Purkinje cells, they're from the brain, but different areas of the brain. Yes, we went through a lot, I know. You have time to review, lots of time to review. We went over the different layers that protect the brain, meninges. We went over the ventricles, meninges and ventricles of the brain, and we went over the different parts of the cerebellar cortex, all the different white matter tracts, and some types of neurons that we find, like pyramidal and Purkinje cells, and how someone, even though with severe brain damage, can survive something like this, because this is damaging something that's not vital for survival, but important for things like decision making, things that make up your personality and basic intellectual processing. Please take a moment, read over this. Any thoughts? Okay, so we have improper drainage of the cerebrospinal fluid. What could cause improper drainage? Well, for cerebrospinal fluid to drain properly, we need those arachnoid villi to be draining into the dural venous sinuses. So if you have increased pressure on blood vessels, if you're blocking blood flow, that could sorry, what am I trying to say? Proper drainage could lead to which? It could lead to perhaps the ventricles. Accumulation of waste products. Increased blood pressure. Yeah, C. It would be C. So if you do have improper drainage, if you're building up pressure in your brain, increased pressure can block blood flow. Increased pressure or increased amount of fluid means you have a lot of waste. Increased pressure could mean headaches and brain injury. The ventricles wouldn't collapse, they would expand. Ventricles would expand. There's another question. Okay, if you have damage to any of these areas, which one's the most serious in terms of your basic function, survival? Prefrontal cortex, frontal lobe, that's cerebral cortex. That's what Phineas gauge damage, so it's not that. Cerebellum, important for balance, but not important for survival. Thalamus, that's important for relaying sensory information. Again, important, but not important for survival. Pons, you're gonna have your most basic functions here in the pons and medulla and midbrain. So the answer is D. Okay. Let's go over some brain stuff. I am going to show you things on these different models again. I've only pointed out some things, but I'll point out more. what's all this stuff? Right. I'm following along. If you want to follow along, you should get out the brain pinning thing. This is from Lab Exercise 14, Chapter 15 Study Guide. So I'm following along here with all these terms. Specifically, if you go to where it says brain pinning, I'm following along that. I'm going to point out all those different things on the models and on the actual brain. Okay, let's start. Let's start with this one. So we're looking at a whole brain with the outer covering, with the meninges. The hardest part that you see here, you can see the external occipital protuberance. This is bone. So this is cranium. This, the whitish part, that's dura mater. Arachnoid is this, the clear one. So arachnoid, dura mater. This is a dural venous sinus, and these are arachnoid villi. Arachnoid villi. And you can see the dural venous sinuses going all through. Please stop me if you want me to go over something again. Anything that you want to see again. Right. Sorry, I'm just cleaning up a little bit. There's a lot here. Okay. So those are the meninges. The ventricles, we can see the ventricles on several models. Here's one model where you can see them. Once again, lateral ventricles, interventricular foramina. Interventricular foramina, it's what's connecting the lateral ventricles to the third ventricle. So from an anterior view, the third ventricle is just a very narrow thing, but from the side, it's more circular. This is the third ventricle. Third ventricle has that hole in the middle. That hole is for the interthalamic adhesion. It's for that connection between the two thalami. Cerebral aqueduct, fourth ventricle, and then from fourth ventricle, central canal. The pink, you can find pink in the lateral ventricles. You can find pink in the third ventricle. You can find pink in the fourth ventricle. That's referring to the choroid plexus. That's where CSF is produced. So CSF is produced in all these ventricles. So this is our space-fill model of the ventricles. Whole brain. Longitudinal fissure. Longitudinal fissure. Lateral sulcus. Lateral sulcus. Transverse fissure. Pink specifically producing cerebrospinal fluid in that previous model. Pink is producing the cerebrospinal fluid. So it's the choroid plexus made of ependymal cells producing cerebrospinal fluid. Transverse fissure. To differentiate between frontal, parietal, occipital, and temporal lobe, well, temporal lobe is easy. Look for the lateral sulcus. So this is temporal lobe. To find the frontal lobe, you need to find the central sulcus. To figure out where the border is between frontal and parietal, you need to find the central sulcus. it's easiest if you look from a mid-sagittal view. You want to find a little more than halfway back, you want to find the deepest sulcus. So here's the deepest sulcus. So this is the precentral gyrus. Here's the postcentral gyrus. This is the central sulcus. So look for the deepest sulcus, a little more than halfway back. That's dividing anterior, precentral gyrus and postcentral gyrus. This model here nicely delineates it for you already, so you can see from an anterior view, from a lateral view. In red, that's the precentral gyrus, which houses the primary motor cortex. The postcentral gyrus houses the primary somatosensory cortex. Temporal lobe houses the auditory cortex. Occipital lobe houses the visual cortex. If it's a something cortex, that means it's the area where that thing is processed. Visual cortex, auditory cortex, motor cortex, somatosensory cortex, just an area where things are processed. And it's cortex because it's the outer layer of the brain. Okay. Looking at a whole brain. We're looking here. This is an anterior view. Here's an inferior view. The smelling cortex. Yeah. In this model, because they made the arachnoid mater clear, and they removed most of the cerebrum. If you peel away, if you peel in between the lateral sulcus, then you'll see more cortex. That's what that is. It's more cortex. It's the insular cortex. That's the insular cortex. That's where we can process things like smell, among other things. Insular cortex. Here's a lateral sulcus. Deep to that is the insular cortex. Okay. Let's go over, right. So we're looking at a brain. Here's the anterior view. We're looking on the underside. Here's an inferior view. Some things that are easier to recognize. Okay. See these bunny ear things? These bunny ear things, if you look at from where it is in your head, it's just right above the ethmoid. It's right above the ethmoid. Yeah, it's right above the ethmoid. It would be, here's your ethmoid. It would be right here. Those bunny ear things would be right here. So they're important for smell. When we initially smell things, the neurons go to here. This is the olfactory bulb. The very tip of the ear, is the olfactory bulb. Can anyone tell me the neurons that extend to this olfactory bulb, what are they going through to get here? They have to go through the skull. So what feature are they going through to get to the olfactory bulb? What feature are they going through to get to the olfactory bulb? The bone feature. Anyone? Anyone? How bad is this lag? Selaturcica is where the pituitary sits. This is where the infundibulum would be. See that hole? That's where the infundibulum would be. The selaturcica would be over here. So that's a little too far back. Talking about over here. Yeah, it's the olfactory foramina of the cribriform plate. Olfactory foramina of the cribriform plate. The neurons. Here's your nose, nasal cavity. Olfactory neurons will go through the bone to the olfactory bulb here. Through the cribriform plate, the olfactory foramina of the cribriform plate to those the olfactory bulb. All right, so you have your olfactory bulb here. The bulb is where you find neuron cell bodies. From the bulb, we then extend back posterior caudal. These are axons. So the tip is the bulb. This is the olfactory tract. Olfactory tract. So these are axons extending back, and they'll eventually make their way to the insular cortex. So olfactory bulb, olfactory tract. That's actually, so we're going to get to the cranial nerves later. That's the first cranial nerve. That's the first cranial nerve. The second cranial nerve are the optic nerves. You can see one here. You can see the other here. We have two eyes. The eyes would be right here. And you got one nerve from one eye, so it's been cut. One nerve from one eye, one nerve from the other eye. And they cross. That crossing point is called the optic chiasm. C-H-I-A-S-A-M, optic chiasm, that crossing point. And remember, it's that crossing point that gives you a hint of where the hypothalamus is. Optic chiasm, hypothalamus. Okay, so optic nerve, optic chiasm. When we continue past the chiasm, when we continue the X, it's now called the optic tract. Just like here we had olfactory bulb and then olfactory tract, and the olfactory nerve is what's going into it, into the bulb. So it goes olfactory nerve, olfactory bulb, olfactory tract. We follow the same pattern here. Optic nerve, optic chiasm, optic tract on either side. Olfactory nerve, olfactory bulb, olfactory tract, optic nerve, optic chiasm, optic tract. And the optic tract, again, is where just above it is the hypothalamus, and here, this hole, this is where the infundibulum connecting to the pituitary would sit right here. The pituitary has been cut off, so you don't see the pituitary here, but that's where it would be. On this model, you can see the pituitary. Yeah, you can see the pituitary, you can see the optic chiasm right here. This is the optic chiasm, and then here's the hypothalamus. The medulla, let me just show you a few more things you can see here. I'm going out of order. Okay. So there's a lot of nerves here. There's 12 nerves. We're going to go over all of them later, but I've gone over one. That's optic. That's olfactory. Number 2 is optic. The fifth nerve, nerve number 5, is identifiable on this model and on the brain. See this really thick one here labeled number 5? This is the pons, and it's on either side of the pons. So this thick one here, this thick one here. That's cranial nerve number 5. It's called the trigeminal nerve. Cranial nerve number 5 is the trigeminal. It's just lateral of the pons. Lateral of the pons, right? See, it's labeled number 5. Lateral of the pons. It's the biggest cranial nerve. Trigeminal. We'll get to why it's called that later on. When we look at the medulla can be, I mean, it's got lots of parts. It has all these nerves coming off of it. We'll get to that later. But it's got 2 midline parts and then 2 bulges on either side. See this round bulge here, labeled 38? And then there's another bulge on that side. So you got 2 round-shaped bulges here, and then 2 midline main structures in the middle. These midline structures, they're known as pyramids, medullary pyramids. Those midline structures are pyramids. Those round structures are the olives. Olives are gray matter. You find nuclei there. Pyramids are white matter. You find tracts there. So once again, we're looking at the ventral side of the brain. Here's the front of the brain, anterior. So this is ventral. The round bulges are olives. The midline things are tracts. They're pyramids. Olives, pyramids. Going to a brain that has frontal cuts. When you start cutting in the brain, you'll start to see a pattern. You start to see patterns. The gray matter on the outside, remember that's called cerebral cortex. Cortex means gray matter on the outside, following the grooves, following the sulci, the gyri and sulci. But then you start to see gray matter towards the inside. This gray matter that's kind of lateral of the lateral ventricles, this gray matter here, there's different names for the different parts, but all together they're known as the basal nuclei. You may hear it called basal ganglia, but that's not the right word since ganglia is a peripheral nervous system thing. These are basal nuclei. You might know basal nuclei if you're familiar with Parkinson's disease. In Parkinson's disease, it's this area that's getting degraded. Like this gray area here and this gray area here. All together, it's the basal ganglia. That starts to deteriorate, and so these areas are important for motor inhibition. It's like along with the cerebellum, it helps with steadying your motions. So it's important for your motor control. It's not generating your motor control, but it's fine-tuning it. Your primary motor cortex is for generating the control. Basal nuclei help fine-tune your movements. This squiggly thing here on either side, it's called the hippocampus. You only see like this squiggle here and this squiggle here, but the hippocampus is actually a really big area. This is a frontal section. Here, this is a model of interior parts of the brain. You can, hopefully this is recognizable. Here's the anterior side of the brain. Here's the corpus callosum. Here are those olfactory bulbs right here. But where it gets smooth here, not including this round bulge, but right here, that's the hippocampus. Hippocampus in Greek means seahorse, so it's seahorse shaped. So not this. This is the corpus callosum, but this. This is the hippocampus. And so when you cut it in a frontal cut, you see this. That's the hippocampus. The hippocampus is really important for lots of things, but two things that you know of. It's important for spatial memory. So me knowing how to get my way around town or even in my own house, just knowing where things are. Spatial memory, locations, and hippocampus is important for that. And if you've seen movies like Memento or 50 First Dates, people forget things really quickly. They can't form long-term memories. The hippocampus is important for that too. Yeah, so hippocampus is important for spatial memory and long-term memories. So like what's her name? Drew Barrymore in 50 First Dates, she damaged her hippocampus. It's a true thing. There are patients like her. It's actually pretty sad since they can't remember things and they're always freaked out because they can't remember things. Okay. Oh, also you can see the insular cortex in this cut. See, here's the lateral sulcus. And so this gray matter, this is the insular cortex right here. So insular cortex, basal nuclei, hippocampus. The hypothalamus would be, where are you, hypothalamus? Yeah, the hypothalamus is just at the very, very base, just like the small gray matter over here on either side. Hypothalamus, basal nuclei, hippocampus, insular cortex, cerebral cortex. Anything you want me to repeat on those things? Okay, we've done the anterior brain, or sorry, we've done the cuts of the brain, we've done the underside of the brain. Now let's look on that mid-sagittal cut. Give me 1 second, I need to grab it. Okay. Mid-sagittal. Anterior, posterior, superior, inferior, rostral, caudal. This is the corpus callosum. Corpus callosum. The corpus callosum, if you go from posterior to anterior, it hooks back. It's pointing at the anterior commissure. Here's the anterior commissure. I hope your kids are treating you well, Andrea. Okay. The lateral ventricle is right here, and so separating the lateral ventricles, that would be the septum pellucidum. And I should have put the other one away. Go. You can also see the septum pellucidum separating the lateral ventricles here. So the septum pellucidum is the wall separating the lateral ventricles, and so that you would see it here, and we're going to see it in our actual brain. What's next? So lateral ventricle, third ventricle would be in this spot, cerebral aqueduct, fourth ventricle, central canal. The space is the third ventricle, but the actual brain tissue, that's the thalamus. So the actual brain tissue on either side is the thalamus, and the connection is the interthalamic adhesion. Interthalamic adhesion. Okay, if this is the thalamus, go back a little bit. This is the pineal gland. This is where you'd find the pineal gland. This little protrusion out right here. Pineal gland. Pineal gland, thalamus, hypothalamus. There would be a connection, there should be a connection from the hypothalamus down here to the pituitary. This is the infundibulum, pituitary is down here. I should probably just show this. Infundibulum, pituitary sitting inside of sella turcica. Okay. If you look, scientists are funny. If you look at the underside of the brain, optic chiasm, this is where the infundibulum would be for the pituitary. And then see these two round things? They look like breasts, I guess, because they're a pair of round things. They're the mammillary bodies. These two things are mammillary bodies. We'll get to their function later, but that's what they're called. They're called mammillary bodies. You can see them also from a side view. Optic chiasm, infundibulum, mammillary bodies. Again, optic chiasm, infundibulum, mammillary bodies. You can see it on this model too. Here's the olfactory bulbs, corpus callosum, hippocampus. Oh, Okay, I'll get to that in a second. Here, it's missing one, but you can see it on this side. Here's a mammillary body. The mammillary body connects to a tract. You have to follow my pointer carefully. Mammillary body following the tract this way. This one. I know there's another tract there. I'm not pointing to that one. It's this one. That's called the fornix. Fornix. So the mammillary body connects to a fornix, which isn't shown very well here, but it would be connecting this way. Fornix. What was I going to show you next? Oh, right, this one. One other thing that you can see on this model, besides the olfactory bulbs, corpus callosum, hippocampus, mammillary body, fornix, this big round thing here, it's almond shaped, I guess. The word for almond is amygdala. So fornix is F-O-R-N-I-X. Amygdala is M-A-M-Y-G-D-A-L-A. The amygdala is important for fear learning. So like if you associate a tone with a shock, like that classical conditioning where you torture poor mice, that's the amygdala. Amygdala, hippocampus, and then around the corpus callosum, the tissue, this region around the corpus callosum, it's called the cingulate gyrus. Cingulate gyrus around the corpus callosum. That area is important for general emotions, which means it's also involved with like things like reward. So it's also involved with addiction. Cingulate gyrus. Cingulate gyrus would be around here. Hippocampus, amygdala. Altogether, those three things, hippocampus, amygdala, cingulate gyrus, they're called part of something called the limbic system. Limbic system is cingulate, gyrus, hippocampus, amygdala. Very generally important for emotion and memory. A few more, and then we're going to the real thing. What's left? Medulla, pons, midbrain. I'll show you all that. Okay. So from the side view, from the sagittal view, medulla is the narrow part. Pons is the round bulge. Midbrain is right here. Remember, the cerebral aqueduct goes through the midbrain. So this whole region is midbrain. If we look on the back of the midbrain, the dorsal side of the midbrain, you see these two bumps? Those two bumps. Together, they're known as the Corpora Quadrigemina. I spell that right? Yeah. Corpora Quadrigemina, literally four twinned bodies. There's two on one hemisphere, there's two on the other hemisphere, so there's four bodies. Corpora Quadrigemina. The ones on top and the ones on bottom, you can refer to them separately. The ones on top are the superior colliculi. Colliculus means hill. So the superior ones and the ones on bottom are the inferior colliculi. So together, the superior and inferior colliculi together on both sides make up the Corpora Quadrigemina. Their basic importance has to do with startle reflexes. Like if you hear thunder and you jump, or if you see lightning and you jump, it's mediated by one of these areas within the Corpora Quadrigemina. Startle reflexes, Corpora Quadrigemina of the midbrain. Arbor vitae within the cerebellum. I think that's all of them. That's it. There's a lot. Any questions that I can show you on models before I transition to before I transition to brain, like real brain questions. You all still with me? Yeah? Yeah? Let me know. Okay. Sorry, give me one more second. Just setting something up. Okay. Sorry, I'm still here. Y'all still here? Are you going to need to know what the parts? Are you going to need to know what the parts do? Yes. And we're going to go over that on Mondays, in Monday's class. We're going to go over what all these parts do. For now, since this is lab, I'm just pointing them out and interspersing what they do. So I'm going to keep the screen here. I probably need to turn the lights down, maybe. Hopefully that helps. All right, let me point out some things that we see. Gloves, can you all see this? Where are my gloves? There are my gloves. All right, so here we have a mid-sagittal cut. We have a mid-sagittal cut of a human head. The brain has been removed. The brain has been removed, but you can see some really cool things here. You can see, how do I put this? Dump this fluid out. You can see this little hole here. This is where this is the cella turcica. This is where the pituitary would sit. There's a little passageway. Here's the nasal cavity. And so the olfactory foramina would go up here, and the olfactory bulbs would be found up here. You can see just a small piece of the falx cerebri. I'll show you a better picture later. This piece of dura goes in between the cerebrum and cerebellum. So the cerebellum would be here. Here, the cerebrum goes in here. You can see some of this dura. It's very, very tough. Very, very tough material. And here's a blood vessel. This is a dural venous sinus. This is a dural venous sinus. Everyone can hear me okay? I'm away from the computer, so I can't see the chat immediately. If you want me to change the picture, make it brighter, make it darker, let me know. Still playing around with how good I can get the picture. Cool, thanks. Okay, so that's what we can see just in this half cut skull. Put this away for now. Let's take a look at the real brain that's been taken out, and we'll look at the meninges. This is anterior, this is posterior, superior, inferior. So here's an inferior view of the brain, anterior, posterior. posterior over here. You can see the brainstem. Medulla is right here. Here's the medulla. This thick outer covering, that's the dura mater. Thick outer covering is the dura mater. I'm scratching it, nothing's happening. Very, very durable. Pretty thick. If you peel the dura away, too close. If you peel the dura away, then you start to see, you see how the wrinkles of the brain are kind of covered? On autofocus. See how the wrinkles of the brain are covered? That's arachnoid. It's a very thin, flimsy covering of the brain. Here we go. This is arachnoid matter. If you peel the arachnoid away, then you can start to see the grooves, the gyri and sulci better. And we can just assume that pia is directly against the brain. See how you can see the sulcus now? Before this was covering it up. Those are the meningeal layers. With all this protection, why are concussions so common? Well, I mean, you can only protect something so much. Concussions are when you hit your head really, really hard. So, I mean, I can hit my head like this and everything's fine. But if I get a huge blow to the head, that's, you know, you can only do so much. Just like seatbelts can only do so much. Just helmets can only do so much. They can only do so much. What else can you see on this? So this midline is a longitudinal fissure. You can actually see the dural venous sinus here. You get the focus. You can see the sinus right here, this midline here. Then if I were to pull this off a little bit, you can see the falx cerebri. So when it fuses together, the falx cerebri is in there. Here's a brain that's already been cut. You can see part of the falx cerebri right here. This is the falx cerebri. Here it's been removed. In this region it's been removed, but here's that corpus callosum. The falx cerebri will go all the way around where the cerebrum is. Down here, all the way up and around. It would not cover this. Because here's the corpus callosum. Commissure fibers connecting the hemispheres. So it's not going in between. Around here. What's the next thing? Next thing I want to show you is, all right, so if the falx cerebri is going to the longitudinal fissure, what's another fissure? This is the transverse fissure between the cerebellum and the cerebrum. That's a transverse fissure. There is dura between it also. There is dura mater in between here. The sulci, this is the lateral sulcus. So here's the frontal lobe, here's the temporal lobe. Occipital is back here. If we want to find the division between frontal and parietal, we need to find the central sulcus. How do we find that? It's easiest when you're on the mid-sagittal cut. And you want to go back more than halfway and find where you find a deep sulcus. So here's a really deep sulcus. This is the border. This is the central sulcus division between the frontal lobe, which is very big, frontal lobe, parietal lobe, and then occipital is back here. frontal, parietal, occipital. Don't worry about the division between parietal and occipital. I do want you to know the division between frontal and parietal. That's the central sulcus. So here's the precentral gyrus. Precentral gyrus houses the primary somatosensory cortex. Postcentral gyrus, postcentral gyrus houses. Shoot, I think I said it first. Let me start over. Here's the central sulcus. The precentral gyrus houses the primary motor cortex. Here's the postcentral gyrus, which houses the primary somatosensory cortex. Precentral is motor, postcentral is sensory, somatosensory. That's correct. Yeah. Okay. Any questions on those meninges, or do all brains have the same exact folds? Do they vary a little? For the most part, like the ones that I'm discussing, they have the same folds. We all, if standard average development happened, we all have the longitudinal fissure, we all have the transverse fissures, we all have the central sulci. The amount of wrinkling, or how many exact wrinkles there are, can vary a little bit, but the major ones are still there, which is why we're just going to go over the major ones. Here we're looking at the underside, inferior aspect of the brain. Some things that we can see here. See this little bunny ear thing? There's another one right here. These bunny ear things, sorry, I only have one hand here. These bunny ear things, these are the olfactory bulbs. Olfactory bulb, olfactory tract. Olfactory bulb, olfactory tract. See the olfactory tract right here. Olfactory bulb. Bulb tract. Oh, yeah. Okay. See the crossing here? Here's one optic nerve. The other optic nerve has been cut. So optic nerve, cut optic nerve, optic chiasm. Here's the optic chiasm. You look underneath all these blood vessels. I don't know how well you can see it, but I can see it from here. Actually, you can see it right here. See that white one? This is one of the mammillary bodies. This bulge right here is one of the mammillary bodies, right there. Mammillary body. Optic chiasm. This is where the infundibulum would come out. You see, I can even stick it in the brain. This is where the pituitary should come out, right here. And then mammillary bodies. You can see the trigeminal nerve. Here's the pons, the big round thing. Here's the pons, the lateral of the pons. See this? When I'm pushing back and forth. That's the trigeminal nerve. Just lateral of the pons, largest cluster of nerves together. Trigeminal nerve. On the other side, it's this thing right here. Trigeminal nerve. So this is the pons. This is the medulla. See this nice round bulge here? That's the olive, medullary olive. And the things that go midline, those are the pyramids. Pyramids, olive, the round thing here. See the cerebellum, of course. Let's go to, go back to this. Okay. So in this mid-sagittal view, again, we can see the falx cerebri. This is falx cerebri. Here's the corpus callosum, very identifiable. Use this to orient yourself. This one's actually a bit smushed. Let me switch to the other one. This brain is not showing the septum pellucidum very well. Let me find a different brain. Please bear with me. Another brain. One. Okay, on this brain you can see how soft this is, how flimsy this membrane is. That's the septum pellucidum. Septum pellucidum leading into the lateral ventricles if you cut it. Corpus callosum, septum pellucidum leading into the lateral ventricles. The third ventricle is right here, this space. The whole circular space. And the little protrusion here, that's the interthalamic adhesion. The actual brain matter is the thalamus, but the space that would be fluid filled surrounding the circle, that's the third ventricle. Follow the third ventricle, you see this little passageway? That's the cerebral aqueduct. Third ventricle, cerebral aqueduct, fourth ventricle. Lateral ventricle, you can't see the interventricular foramina. Third ventricle, cerebral aqueduct, fourth ventricle, leading to the central canal. So here's the central canal going through the medulla onto the spinal cord. Central canal. Fourth ventricle, the central canal. Those are the ventricles. Okay, some brain regions. Let's go to some brain regions. Okay. Surrounding the corpus callosum, this region, immediately surrounding the corpus callosum this way, that's the cingulate gyrus. Here to here. Cingulate gyrus. Corpus callosum. The corpus callosum, follow the hook. It points to a dot. See how this circular area is different from around it? These are axons. This is the anterior commissure. Here's the pointy part of the corpus callosum. This is the pointy tip of the corpus callosum. Follow the tip for a round thing that's fibrous, because it's axons that have been cut. This is the anterior commissure. Here's the thalamus. Go posterior to the thalamus, look for a raisiny looking thing. This raisiny looking thing is the pineal gland. And then inferior to it is the hypothalamus. So thalamus, pineal gland, hypothalamus down here. It's a little broken up here. On this one, you can actually see the hypothalamus better. Yeah, you can. Okay. So here is thalamus, hypothalamus. And you can even see the optic chiasm right here. Here's the optic chiasm, hypothalamus, thalamus. Here's the midbrain, cerebral aqueduct. Those two bumps are the superior and inferior colliculi, the corpora quadrigemina of the midbrain. The corpora quadrigemina are part of the midbrain. Round bulge is the pons, narrower is the medulla. You can see the arbor vitae in the cerebellum. If you're lucky here, if we look inside of the lateral ventricle, if we look inside of the lateral ventricle, you can see some tissue here. That's choroid plexus. It's dark tissue. It's because it's surrounding blood vessels. That's choroid plexus. That's how CSF is produced. I think I've gone over everything that I can show on the brain. It's 153. After I can show more things. So if there's something you want me to point out again, let me know. But I'm probably going to make a recording later this week that's dedicated just to pointing stuff out on these models in the brain. So it's not as long, it's more concise. But I'll give you a minute. Let me know if you have any questions. Oh, gross. Questions? There's a thalamus. More accurately, Andrea, is the thalamus in the third ventricle? I would say the thalamus is surrounding the third ventricle. So here's the third ventricle. Lateral view with the dot through it. Anterior view looks narrow. So the third ventricle is like the cream filling. The thalamus is like the cookie of the Oreo. So thalamus is on either side. Third ventricle is in the middle. Any other things I can, what's the word? Any other things I can address? Okay, if you have more questions, I'm checking my email constantly. Please contact me. If you want a video chat, we can do that too. Probably I can probably do it Friday. Friday, I will work on getting videos that are just specific to these models and to the brain. My brain's a little fried from talking so much. It's sad without people. But I guess I have Napoleon Bonaparte. Someone get me a little coat for him or something. That'd be nice. Otherwise, he's just got a nice mane of hair, I guess. How How's the practical going to work? Just like this mini quiz, you got pictures and arrows. It's going to work just like that. There's still an activity. It obviously has to be kind of multiple choice. But basically, I'll give you like a ton of choices, like a ton, like 20. And I'll ask you to point to an area with this, point to an area with that. For this practical, it's going to focus on muscles. So think about what name of muscles, location of muscles, action of muscles, and like what joints they act on. Think about that. Just a reminder of what I'd like you to do now that you've hopefully watched all of this. Now that you've hopefully watched all of this. You have a quiz on Monday. All of you do. Monday, Wednesday lab. Your lab check 8 and activity 7 is due on Monday. Tuesday, Thursday lab. It's due on Tuesday. I will go over these things again in a separate video, more concise. I just need someone to help me tape this, so I'm going to get some help. And when I'm less tired. Cool. Thanks, everyone. I will see you all later. Stay safe. Take care.